Okay, biomedical innovation students. I'm going to talk to you about Friday, Monday. So this is for January 8th and January 11th, right? So January 8th and 11th. So let's talk about the first thing. Continuing with the lab. I'm not going to be having you do really anything much outside of class related to the lab, except at least for this session between now and Monday, because we're waiting on the samples. If you remember where we're at in the activity, we have just hopefully ligated in our reaction to the DNA samples. And then the next step in the process then is to do gel electrophoresis. And the goal of that, kind of look at the whole overview of the process. Our goal here is to confirm that we had ligation. Ligation actually took place. So we have taken our reaction tube, which has the ligated DNA with, remember, both the C1, the R1, both the C1 tube and this initial stock ligation mixture had pieces of the kanamycin gene and the plasmid in it. So basically, they had these pieces in them, right? They had this and this. Actually, had the plasmid cut, cleaved vector, and then the gene. Okay, so then additionally, T4 ligase was what was in the green tube, and so then we added the DNA pieces to that green tube, the T4 ligase tube. Hopefully, during that 60 minutes, they were sticking together and making a new plasmid. So now we're going to confirm that by comparing the size of our control group, which did not get any of the T4 ligase, and we're going to look at the size of that on a gel compared to the size of the reacted gel sample. And so we'll run that through and do the electrophoresis. That will not take the whole time on Monday. I'm then going to ask you to go ahead and look at Module 2 because we're going to finish Module 1 and start Module 2 on Monday. So when we finish up Module 1, basically we'll doing 3 lanes. This is our standard marker, our ligation control, and our ligation reaction. And this standard marker is going to be more important than normal because we're actually going to use it to estimate the size of the fragments that we have. And then the staining process, we'll talk all about the procedural stuff. We're actually going to use the CyberSafe stain, so we're not planning on using this type of staining anyway. And then Module 2 we'll get to, but we're basically going then to try to transform our bacteria from our plasmid, and so we'll do the first step in that on Wednesday. Yeah, you'll see you guys on Monday and Wednesday. So we'll do the next step of that on Wednesday. So because we do have to let the gels sit and analyze them, so we can't do that until we've seen them. So anyway, so that takes care of kind of where we're at in the lab. You will eventually, I don't think you need to work on it yet, do the Module 2 organizer, but I'll tell you to do that more on Monday. So really don't worry about anything with this lab at this point. Make sure you've turned in your restriction enzyme problems. So while that's going on, we're also going to be working in mission file 2 or problem 2, which is exploring human physiology. So I'd like you to, for Friday, before you come Monday, read through the human physiology mission file. All right. And you'll hear about this story of Alex Marion and some of the complications he's been having with a condition called acute intermittent porphyria, which has to do with deficiency of an enzyme called PBGD, and ultimately results in a lack of oxygen being carried in the red blood cells. So in this activity, your mission is to learn more about the research process, because if you're going to treat, diagnose more effectively conditions like Alex's, scientific research has to be done. So you're going to pick a topic of interest related to human physiology, and that's going to relate to like the function of one or multiple body systems. So you can think about testing the respiratory system or the cardiovascular system, test the nervous system with reflexes or something like that. Keeping in mind COVID and being able to do it safely without risking transmission or increasing interaction. So we're first going to look at scientific research and background and do like a study design of it, like you're going to design a study. And then we're not going to do all this. We'll do some statistical analysis because you have to do enough trials to provide a statistical significance. And so we're going to get into the math of what that means. It's called a t-test. So we'll get into that next week, a week after. Ultimately, you're going to make a poster presentation for your experiment. So this experiment will take a few weeks for us to do. Hopefully, you'll use some sort of technology or some equipment like heart rate sensors or oxygen sensors, something like that you can use. So we'll go through some potential ideas for that in class. So what I want you to do for Friday, after you read through the mission file, is in 211 that talks about scientific research. You're going to do an investigation of the different research study designs. So if you go down here to click on this document that says the research design interactive presentation, and that should open a PowerPoint. And just in case it doesn't, I'm going to make this a Google Doc and put it here in problem 2, just so you have it there as well. So you'll open that and then go to Canvas under problem 2, which I've now moved under problem 1. I put it under problem 2, scientific research. And when you open this doc, see this, and this is what you're going to fill out while you look at that presentation. And so you can see what that presentation looks like. And it talks about experimental studies and observational studies, which are the main 2 types of research design. And you can see how kind of give an overview of those 2 things to start with. So you're going to look at the description of each, when they're conducted, pros and cons. Does it lead to, do you get causal or correlational relationships from that? And so you'll look at more information on that as it goes. So it gives quite a bit of information on that to begin with. There's 39 slides, so this is not something I expect you to have done necessarily when you come to class Tuesday. The goal is to be done with this by the 13th, which would be Wednesday. So you'll have, really, this is kind of your outside of class work that you'll be doing, addition to the Module 2 organizer section that you'll be working on both really Friday and for Tuesday. And then hopefully, I don't know, well, I don't say hopefully, once things are safe enough, we'll be back in person full time. We can move things a little more quickly. But you'll also look at more of the specifics in that of the types of observational studies. And then you'll look at the experimental studies and some of the information with that. You can click through like this. Some of these have like questions as well. So if you wanted to play the slideshow, then those hyperlinks would work for you as well. So any questions about, or if there's any questions about that, make sure you email me and let me know. But basically use this slideshow to help fill out this document. And that document is not due till Wednesday. So have a great weekend and email me if you have any questions, and we'll continue with the lab on Monday. Thank you.